Hello, and thank you for viewing my talk as part of the Global Coral Reef Week virtual conference. My name is Khen, and I'm a PhD candidate in Dr. Jennifer Smith's lab at the Scripps Institution of Oceanography at UC San Diego. My presentation is titled 10-Year Time Series of Coral Reef Benthos on Palmyra Atoll Shows Evidence of Long-Term Stability. I'm also a digital scientific illustrator, by the way, and throughout this talk you'll see plenty of my own drawings. To start off, as I'm sure you're all well aware, currently with the progression of global climate change, more coral reefs are headed toward degradation than recovery. And arguably, we need more studies that precisely track how reefs are changing through time in order to better understand their responses, both current and what may happen in the future. This illustration of mine shows the transition from a coral reef dominated by healthy corals and other calcifying organisms such as crustose coral and algae, to one dominated by turf and other fleshy algae, as well as corals that are diseased, bleached, or dead, which fundamentally cannot provide the same ecosystem services for which coral reefs are known and valued. As a side note, in case you're not familiar with this term, benthos refers to the community of organisms that live on, in, or near the bottom of a reef. The two main benthic groups that I'm interested in for my research are corals and algae. Corals are colonies made up of individual polyps that contain zooxanthellae, or photosynthetic microalgae, which provide them with most of the food source that they need to survive, and also their pigmentation. When corals are exposed to higher than average water temperatures, they can lose their zooxanthellae in a process called bleaching. They then turn white and are essentially starving, making them more susceptible to disease or death. Algae are highly diverse morphologically, taxonomically, and functionally, and can be separated into three groups. Turf algae are largely fleshy. They're opportunistic and very fast-growing. They can outcompete corals and other algae and tend to dominate after disturbances. They're also a primary food source to herbivores. Crustose coralline algae, or CCA, are more slow-growing on the other hand. They contribute to the carbonate budget of a coral reef, and as such can be considered reef builders and potentially indicators of the reef's health. Macroalgae can be further classified as either calcareous or fleshy, and the abundance of one type over another has different implications for a reef community. The broad overarching goal of my research is to explore the long-term effects of thermal stress on corals and algae. In setting out to do so, I've been fortunate enough to have the chance to study Palmyra Atoll, which is a remote uninhabited island located about 1,000 miles south of Hawaii in the Central Pacific. Aside from brief military occupation during World War II, Palmyra has experienced minimal human impact and is now under a high level of federal protection. It also has no local stressors such as fishing or pollution, and in fact its reefs can be considered quasi-pristine. Altogether, this makes it an ideal location to study the effects of global stressors such as climate change without the confounding effects of local stressors. So first, some background information on the temperature history of Palmyra's reefs in the past decade or so. This plot shows the change in sea surface temperatures over time as measured by both sensors, which were deployed in situ at each of the habitat types on Palmyra, and satellites via remote sensing. When the sea surface temperatures exceed the Palmyra-specific bleaching threshold, indicated here by the dashed black horizontal line at about 29.4 degrees Celsius, the corals of Palmyra are expected to bleach. And this happened in 2009 and 2015, each of which corresponded to a mass coral bleaching event. 2015 had a wider peak or lasted longer. In both cases, this was associated with El Niño Southern Oscillation, or ENSO, which is becoming more frequent and severe. My project is based on a time series of permanent benthic photoquadrats that have been taken annually on Palmyra over the past 10 years from two different habitat types. The fore reef, or FR, which is slightly deeper at 10 meters and has stronger wave action, and the reef terrace, RT, which is slightly shallower at 5 meters, and is more sheltered, potentially more exposed to solar heating and light, and also receives lagoon outflow, so more fine sediment transport. Within each of the 2 habitat types, there are 4 sites. 2 of the FR sites are located on the north shore, FR7 and FR9, and 2 on the south shore, FR3 and FR5, about 5 kilometers apart respectively. All 4 of the RT sites are on the western side, slightly closer together at less than 1 kilometer apart. RT4 and RT13 are nearer to the lagoon and consequently more turbid waters. RT1 and RT10 are nearer to the open ocean. Within each of the 8 sites, there are 10 replicate plots. And the reason that we considered each site individually is to increase statistical power. So to reiterate, photoquadrats were taken on Palmyra at every time point for each site, every 5 meters along a 45-meter long transect from 2009 to 2018. A 2019 time point was taken in September, but has not yet been fully digitized. This figure shows on the left a picture of my advisor, Dr. Jennifer Smith, who established this time series, taking a photoquadrat. On the top right, an example of a photoquadrat, and on the bottom right, a digitized photoquadrat. I'll now take you through an example of just one quadrat's change through time, showing you my digitization methods. Using Adobe Photoshop, we trace the borders of all live coral colonies, algal patches, and any other benthic organisms that we see in the plots. We then fill them in with a swatch color that identifies them to the finest possible taxonomic resolution, genus or species. Then, since we know the actual dimensions of the photoquadrat frame, 0.6 meters by 0.9 meters, we can extract the planar area measurements in centimeters squared based on pixel counts of what we've traced and now filled in. It takes about 2 hours to trace each image, and this can be very tedious. But this is a 10-year time series among the most thorough and longest existing of these types of data sets, especially for a remote reef. And as of this day, we have over 1,000 images total, 1,107 to be exact, and now here they are digitized. This was a multi-year effort with the help of several undergraduate volunteers. I actually think that the collage of the digitized photoquadrats ends up resembling abstract art. But more importantly, what does this all mean ecologically? What stories can we tell from this intricate collection of data? Well, although there are endless possible questions that could arise from this data set about Palmyra's benthic communities, this talk will address just 4 of them. And in terms of timescale, my questions fall into 1, initially or at the beginning of the study, 2, short term, 1 year after each of the bleaching events, 3, long term or over time, and 4, overall after 10 years. So again, first I wanted to see whether the benthic communities at each site were structured significantly differently at the beginning of this study. On the left here is a bar plot showing the percent benthic cover by group at each site in 2009. As you can tell, there's considerable variability between sites. FR3, RT4, RT10, and RT13 had relatively more hard coral cover. RT13 also had the highest cover of turf algae proportionally. FR9 had a lot of crustose coralline algae. FR7 had the most soft corals, and so on. On the right is a Bray-Curtis-based, non-metric, multi-dimensional scaling plot, or NMDS, of the benthic community composition in 2009, based on square root transformed percent cover values. Each point in this visualization represents how one of the 10 quadrats at each site, and again, there were 10 quadrats at each site, was structured in terms of percent cover. Essentially, the closer that the points lie in this 2-dimensional space, the more similar those benthic communities reflected by the quadrats are. Also, the closer they are to a given word, the more abundant that benthic category is in that quadrat. Now we can see that not only was there variability between sites, but also possibly within them across the different quadrats. However, the NMDS is merely a visualization of the benthic community structure, and it's not a valid statistical analysis. So we followed this up using a permutational multivariate analysis of variance, or PerMANOVA, on this same Bray-Curtis distance matrix, which confirmed, as you can see by the p-value highlighted here, that the benthic communities at the 8 sites were structured significantly differently even from the beginning of the study. My second question was about the short term, how the corals responded to each of the bleaching events 1 year later in terms of percent covered. And so these plots show the change in cover for all corals combined at each site 1 year after the respective bleaching events, which occurred in 2009 and 2015. For the first thermal anomaly in 2009, 1 year later, there was a minor decline in hard coral cover of up to around 10% for 3 out of the 8 sites, which are circled here. For the second thermal anomaly in 2015, a year later, those same 3 sites declined in hard coral cover again to a similar extent, and an additional 2 sites also declined. Interestingly, the main sites that responded negatively to both anomalies, RT4 and RT13, are actually the most proximate to lagoon flushing, which brings in warmer water, sediments, and nutrients. The fact that in total more of the sites declined in response to the second event potentially indicates more severe warming, which indeed is consistent with measurements showing that for 2015, the sea surface temperatures on Palmyra were slightly higher and persisted for longer than during the 2009 warming event. My third research question was regarding the trajectories of percent cover of all benthic groups, not just the corals, throughout the entire study period to identify successional patterns in community structure. And in doing so using another Bray-Curtis-based NMDS plot, this time on percent cover data over time, it appears that each site, or each different colored line here, follows its own non-coherent path, making it really difficult to characterize these trajectories not only across the sites, but also for each site individually. But it's worth noting that the community assemblages at each site remain strikingly similar site-wise, or they occupy the same region in two-dimensional theoretical space, if you will. So then the sites have their own unique responses, but perhaps this is to be expected. Also note here that the FR and RT sites fall on opposite sides of the plot, and the 2 sites that somewhat overlap, RT1 and FR9 near CCA, are close together in actual geographic space. Of note, the site that changed the most, FR5, experienced invasion by corallomorph. In summary, despite the past decade of environmental change, which as we know included 2 large-scale disturbance events, the sites have, for the most part, retained their distinctive benthic community structures with time. And then again, statistically using a 2-way PerMANOVA on this Bray-Curtis distance matrix to test for the effects of factors site and time point or date, we find that both are significant, as you can tell by the p-values highlighted here. In the blue R-squared values, you can tell that site explains about 43.3% of the variation in benthic community response, whereas date or time point explains around 5%. There's a significant interaction term between site and date, accounting for 6.2% of the variation, and the remaining 45.6% is unexplained. Still, this means that there's evidence that these sites are changing in different ways over time, which isn't too surprising given that they were different to begin with. But nevertheless, it seems that there is little temporal variation in community composition overall by site. Also, that in this case on Palmyra, site may be a better predictor for benthic community responses than the time of observation. My fourth and final question had to do with how the benthic communities on Palmyra have changed between the initial and final time points, which may seem like a coarse metric, but quantifying the net change through time may provide a better sense of the long-term effects of heat stress for these reefs. So these are bar plots comparing the change in percent cover by benthic group at each site between just 2009 and 2018. And we used Tukey's honest significant difference tests to measure differences among sites for each benthic group. As you can see for coral cover in green, at one site, FR3, it decreased, but only by up to around 25%. In another site, RT4, it increased, whereas for the rest of the sites, there were no significant changes. From our imagery, we also know that FR3 experienced the greatest post-bleaching mortality following both of the events. As for crustose coral and algae in red, at most of the sites on average, the cover was roughly similar 10 years later, hovering at around 0% change, except for FR3, which increased, and RT4, which decreased, leading us to think that perhaps the corals that were lost at FR3 were replaced by CCA, and vice versa for RT4. As for macroalgae and turf cover in blue and purple, respectively, they were similar on average between the two time points for the majority of the sites, though it should be noted that as for macroalgae in particular, they're known to exhibit seasonal fluctuations with time. Now, there are a number of more ecological stories we could pull from these data alone, but in the interest of time, I'll take you to, rather than just the net changes, the percent cover by benthic group at each site over time, for which we see that across the 10-year time series, coral cover actually seems stable, relatively high compared to other more degraded geographic locations that we've heard of worldwide, such as in the Caribbean. Coral cover at the fore reef is at around 30%, and at the reef terrace is at around 50 to 60%. However, there are some deviations from stability directly following each of the thermal anomalies, and especially for the second thermal anomaly. Interestingly, for RT10 in 2015, the cover of turf algae spiked while simultaneously the cover of crustose coral and algae dropped. Nevertheless, in the most basic sense, other than those minor absolute changes which naturally we would expect in any ecosystem, it appears that in general, the benthic communities on Palmyra have remained stable in the past decade, which is highly surprising considering the state of other reefs worldwide, and may be a testament to Palmyra's resistance and recovery capacity. My takeaways from this study include that, one, we need to look at benthic community responses more comprehensively, both in terms of benthic group and time span. Unfortunately, most of the studies on coral reefs look just at coral versus macroalgae abundance at really short timescales, like right before and right after a disturbance, understandably due to logistical challenges. But here, thanks to our extended time series, we could see evidence of stability rather than just decline, which we may have seen in just the short term. So this data set speaks to the importance of longer-term, more detailed ecological monitoring. Two, that the effects of disturbance can depend not only on time as a factor, but at some locations such as this one, perhaps even more so on the community composition of the sites themselves. And three, that understanding why Palmyra has been successful will be important for managing reefs under current and future warming scenarios. Perhaps we can use these data to establish baseline information on the structure and functioning of reefs in the absence of local stressors. And we can then apply that information toward managing other more human-impacted reefs, which are at higher risks of decline. A reverse phase shift is pictured here, the optimistic ideal. Now, whether that's possible, at this point, it largely depends on us as humans. In the future, although there are many directions I can take with this data set, I plan to specifically investigate how different coral colonies responded through time. We know that responses to bleaching can be not only location-specific, but even within locations, species-specific, and further, even within species, colony by colony specific. And I want to recognize that individual variability. By the way, these infographics of colony change through time were made from this time series itself. I also would like to answer the question of what specifically makes Palmyra's reefs more able to withstand disturbance compared to other reefs worldwide. Overall, I'm really looking forward to seeing what else we can discover from this goldmine of a data set, and I feel extremely lucky to be involved in this project. There's a lot of dedication that's gone into it in my eyes, and not just from me. Also from the people who established this time series in the first place and have been revisiting it for now over a decade. So I'd like to end by acknowledging my co-authors, especially my advisor, Dr. Jen Smith. Also my longtime mentor, Dr. Levi Lewis, and the wonderful undergrad volunteers who have helped me with image processing, as well as the rest of the Smith and Sandin labs, and finally my funding sources. I'll leave you with this last illustration of mine of an hourglass, which to me represents how, as humans, we need to take action against climate change now to help keep coral reefs alive before it's too late. I'll take any of your questions in the comments below. Thank you so much for listening.